So we're looking at, just to remind you all, we're looking at E. coli solution encapsulated in a droplet, and we looked at it, what's the dynamics at low activity. So essentially, we see some fluctuations in the shape of the droplet, and within the droplet there are largely chaotic and random flow that's been generated. And if we increase activity, so in this intermediate regime, what we see is this droplet is spinning as a whole, and then you generate this vertical flow in the system, and also we see more noticeable boundary changes. Essentially, that's just because the bacteria are more active, so they're able to push the boundary with a larger force. And as a particle goes to the boundary, it's still pushing and it slides in a particular direction. So that's how you generate these spinning droplets at the intermediate activity. Okay. And as some of you have proposed, if the activity is high, then perhaps the droplet itself will move. And that's what we see here. And as well, when the activity is very high, you see the droplet, the shape changes quite violently here, as one would expect. But also it has a center of mass displacement over time. So it's indeed self-propelled. And this line here is just tracking 2 points on the droplet so that you can see it's also rotating as it deforms and translates. So if you look at the trajectory, okay, we've seen these kind of trajectories before. They look just like active Brownian trajectories as expected. And then, well, you can experiment. Again, you can track their center, as you've seen here on the left. You can track the mean square displacement over time. It has a ballistic to diffusive transition that we know already from any kind of active Brownian type of model. So here at the bottom, we're looking at the perimeter of this droplet as a function of time, so that you can see the fluctuations here. So sometimes you have a huge fluctuation. I think you can see that in the video here. At one point, there's a giant deformation that happens to the droplet. So right there. So that's what you're seeing here. So you have these fluctuations, the droplet. So the point is you can make use of active particles as building blocks to form more complex systems. Comments?
So we have to solve one equation for the E. coli bacteria inside the drop.
So you're thinking about modeling these equations. So if you're doing a mean field model, what you can do is first is inside the droplet, you use the mean field for active Brownian particles, right? You do that. Outside, you have the Stokes equations, the flow equations, and at the boundary, you have force balance is there. But lastly, you have to keep track of the deformation of the droplet. There's some the dynamics of the droplet as well. So now you have to incorporate all of those in order to reproduce, what you see here. So it's now more involved than before.
This is one kind of that you think that droplets within the shape changing.
Yeah. It's changing. The shape changing is induced by activity inside the droplet. Yeah.
If you want to also first of all, by fearing to us, we look for the inside.
Yeah.
So like then we get a cue for the goods of it.
You get a
orientation.
Yeah.
Some kind of English for the whole droplets.
Sure.
And then we put it in the we treat the droplet as an active sand.
Yeah. So if you want to reduce model, yes, then you can figure out roughly what's the speed of the droplet, and if you at that level, then you just replace everything by active Brownian particles. That's fine. But yeah.
Yesterday you studied that two each side. There is a cardinal.
You mean the alignment of two ellipsoids? Yeah, that's fine. Yeah.
That will change in the.
So are you thinking about more than one droplet now?
Yes, it's two droplets.
Yes, that will be different because first they're deforming, so the shape at each instant in time will be different.
Yes.
Yeah.
So I was wondering,
history has bacteria inside because obviously the shape of the particle double changes. And it also moves around, right?
Yes.
So it is like an active particle with changing shape.
Yes.
So if we just try to model the bubbles, then we need consider the bacterial also instead.
Now, if you just get a bubble, you can get an effective dynamics. And as you said, precisely, you have the active particle that's shape changing.
But actually, the bubble is nothing but the correction of bacteria.
Sorry?
The bubble is actually nothing but the correction of bacteria.
Yeah, it's due to their dynamics to the bacteria, yes.
On that.
Yeah.
So, on the top right, the figure is with the plot of MST, I think.
Yeah, that's the MST of the red trajectory. Yeah, yes.
The MST is fashion from the proportion to C square proportion to T is aging as it is predicted in each phase. Here we are seeing two linear branches which are intersecting.
No, that's the scaling. The dash line, you talk about the dash line. Those are not linear, they're in log scale, so the first one is quadratic. Okay. Second one is linear.
It's square to T, that's what it is.
The dotted lines intersect at a point.
Yes.
No, the dotted lines are just MST, right? Are you talking about the top graph?
The straight two branches.
The dash lines? Those are not branches, those are a slope that's compared to tell you the slope of the dotted lines. The dotted lines are the actual data.
So has there any particular significance of the point at which the
Yeah, that's tau r. So transition from wave-like to ballistic transition. Opposite. Ballistic to wave diffusive transition.
that is the time scale
For reorientation.
transforming.
Yeah, so that's, you can imagine, effectively this guy also when he swims and he will reorient to the random fluctuation.
Orientation time.
Yeah,
Effective orientation.
yeah. Yes, you can see that here.
Within that orientation time, if we observe within that orientation time, it's ballistic.
Yeah,
And if we have more orientation time, more
yeah. That's just ABPs, okay? It's the same thing. Yeah, that's right.
with tau effective will be different from the ABPs because
Yeah, this is an emergent tau, an emergent swing speed. Everything is emerged from the fact that the little bags are pushing around the
we
top.
can also think it,
Yeah.
now we can think about what should that we were like bacteria
Okay.
pushing the boundary, and now we want to see the emergent tau effective of the bubble break
Okay.
from the back, so, and we can see it emerge. Something is emerging from.
Yeah. Yeah. So that's an effective dynamics. The point is the effective dynamics of the droplet is just like an active Brownian particle. And it has all these properties that we see before.
High activity just active on it.
Sorry?
So high activity just active on it.
Yeah.
So for intermediate activity, you say that
It's spinning.
stable. It doesn't propel out.
Does not.
It just spins.
Just spins in place.
Yes. If we had something like it spins and propels to the ball, we can do
There's yes. It just spins in place. Right? That means moving like is slightly jiggling due to the noises.
But the point is that in the high activity also the shape changes. Like
It always changes because it's a flexible object.
it doesn't affect the effective time towards still a negative.
Of course, it affects. So it's you have to sample the entire system to find how R. And you can't just neglect that. If the shape doesn't change, it's not going to self-propel.
Not just the shape changing ability at media.
Sure. Yes. Yes. Yeah.
And this trajectory is the trajectory of center.
That's the red one. That's the center mass of the droplet. Yeah. Other comments?
If you just see the circular mass, it's almost like a white light. Here, look at the circle.
Yeah, but you're avoiding reality by not looking at the bigger picture. Of course, that's fine.
Yeah.
Other comments?
So like sector mass is like the delays, the related body.
Yeah, because the shape is not, you don't have an analytical formula for the shape. It's all empirically done. In this case, it's image tracking.
Yes.
You track you take exactly, that's what this is, the image. You can find the boundary through image processing, and then you find the center.
So simulation, how do you simulate the change the shape?
You mean what kind of model you're thinking about?
Say this changing the shape like If you want to simulate this,
You want to describe the shape change, or you want to see the shape change emerge from your simulation?
if you want to describe the shape change, say,
Given, as given. Then you have to tell me how the shape changes.
but can that not be generalized like, well,
There are only two possibilities. One is you prescribe the shape change over time. The other is you solve the equations to get the shape change, right? You can't do as
so the shape change will be a consequence of the equation.
you have the hydrodynamics to figure out the shape change.
So it is like automatic. So we solve.
Yeah, you solve the whole system. Everything is there, right? It's all coupled. Yes, correct. It's emergent. Yeah. But Vasco is thinking about doing something simpler so that we don't do that. Yeah. Good. Other comments?
But then we have to find the relation between the shape change and the fact is only this.
Yeah, I mean, one thing you can do is you decompose the shape change into Fourier modes,
That it.
and you solve some fluid mechanics, right? You prescribe a shape change, you have to figure, tell me then,
Yeah.
I don't care about where it comes from, but you said there's shape changing and then the generous flow, right? That will give rise to their motion. So that's a thing, that's some intermediate model you can think about, okay? And actually, I don't think people have done that. So that's something you can think about, okay?
The big hyperlinearness in that shape like a square.
Yes, mathematically, yes, but you don't want to make a square because people will be upset. How do you make a square droplet?
Maybe you can then extrapolate the I mean to say like triangle square and then we
Yeah,
maybe
Yeah.
real formula.
that's right. Yeah, you can do that. Yeah. Okay, great. Other comments?
Right.
So the theme now is we're thinking about, we can encapsulate active particles in droplets and other containers. One might be wondering, why is that useful? That's useful for, say, delivery purposes. Like in drug delivery, you have some sort of vesicle or container that contains your active drug ingredient. Okay, you can also encapsulate some active particles in there, then which will propel your carrier forward. So that's the point of this kind of experiment. Okay, so then you can also encapsulate these active guys in an actual vesicle instead of a droplet. Okay, something like this. The vesicle is made of a membrane, okay? And you put particles in there. And as you would expect, so in this case, we can consider the simplest case where, say, the membrane is not deforming, it's just a spherical vesicle for simplicity. Then in that case, you have to find some other ways to break symmetry so that the vesicle as a whole can move forward, can self-propel. One way to do that is to bias the density of the particles. So imagine somehow ABPs have a higher density on the right and lower density on the left. What happens to the vesicle?
I need to look like.
And the other thing I didn't tell you is water can pass through the membrane into the droplet, okay, but the bacteria cannot come out.
So we worry.
You go to the right, right? It will self-propel. Okay, make sense? So and the point is, then the question is, how do you produce an asymmetric density in the vesicle? The easiest way is to apply a field, which we've done before. You have an orienting field, trying to align the guys that you have orienting field to the right. Then what happens to the bacteria? They're more likely to be here. And that's what you see here. Here we're measuring the propulsive force you get for the vesicle as a function of this parameter. We've seen this parameter before. That's the strength of the field. And you can get a velocity out of it. So again, it plateaus because at most you get all these guys aligned. You can't do more than that. So you plateau at high field frequency, field magnitude. But the point is you can get a vesicle that's self-propelled. Okay? And there's some other strange things that happens with this. Here's an experiment. I think some of you are talking about this case where here you have bacteria in a lipid vesicle, and these guys are flagellated swimmers, so they are very active, so they can generate. They really deform the vesicle quite a lot. Okay, there's a tube that's being extended out of the vesicle, but they haven't break the vesicle yet. So what's funny here is this flagellated swimmer, okay, by protruding this tube, it makes this vesicle a flagellated swimmer. Okay, the vesicle has a cell body and the tail, and that's what happens in experiment. So this vesicle behaves like a flagellated swimmer, which is funny. So that's what you see here. It's hard to look at. So the dark spot here, that's the part of the vesicle that hasn't been deformed much. And you see there's a little thing here. So that's the flagellated swimmer in the tube. And because of that, this vesicle is going to move. Here, you see the flagellated swimmer is at the front, right? It's dragging along the vesicle. In this case, it's not aligned, so you get this circular kind of trajectory. It looks like a chiral active motion that you see here, and it's going in circles. Like, yeah. Okay, so lots of strange systems that we looked at today. And here's another case of an active droplet. So now we know how to make droplets active, and I'm not going to talk about that here yet. So we just have a droplet here that's being put into a passive pneumatic system. Okay, so here's a sketch of that. When you introduce a boundary, that creates frustrations or incompatibilities between the alignment. So there's a defect that would develop at the front. Now, if you look at that, you have a spherically symmetric active droplet. If you have a spherically symmetric active droplet that's not deforming, it just performs active but random motion, not going in any direction in particular, right? Okay, that's important. But if you put that into this environment is patterned. It has a left-right asymmetry. Okay, then what happens to the droplet?
It's going to be into
So if you have an active particle in free space, it's going somewhere, but it's the direction it's going is random. There's no directed motion of an active Brownian particle. If you put an active particle in an environment like this, in a passive pneumatic, there will be a defect that develops here. So the environment now is asymmetric. Okay, the front and back are different. So what happens to the droplet? So you get directed motion, and that's what you see here on the right. So the red is the trajectory of the droplet in a passive pneumatic, and you can measure its velocity here. So it's
But think like I'm just asked high fears of electrical wave and passive droplets in active pneumatics.
It's the opposite.
It's the opposite.
It's active droplet in passive pneumatics. You can do the opposite experiment, yes. Yeah.
Will it develop different results?
Yeah. But also you can imagine, we talk about the tracer diffusion problems where you have a passive particle in an active suspension. So if you're thinking about an active pneumatic and then you embed a passive object, the passive object is going to also pick up activity. Yes. But it will be slightly different from what you see here.
It's giving a little bit of disorder for in the system, right?
Yeah. You have a defect now at the front. So I think here's a.
But it's still passive pneumatic, like
It is passive pneumatic. The active droplets, the activity is encapsulated in the droplet. Yeah. Wait, is this a video or not? It's not playing.
isn't.
So it's going to the right-hand side. So you can control motion of active particles by changing its environment. Okay, so in this case, you have a change the direction of.
This is what the direction by our choice. Technically, that
You could, yep.
shit. Because we what like is in the
Like you can realize what every particle to go in certain direction.
Yeah, that's right.
And if that is can be applied, then we can put an active particle
Yeah.
say,
Yeah, so essentially you want to, there are two things. One is, well, okay, there's two ways, right? One is the particle is smart, then he knows where to go. The other is it's not, so you have to tell where this particle goes by using environmental cues, whether the substrate or some external field and whatever for that to happen. Yeah. Other comments?
Like these represented droplets that you progress, presumably
Yes.
going at the center, see like right unit areas.
Oh, that's just the imaging. Those are the bacteria, the things that are, that's it's a video. So, yeah.
It is not a fluid region.
There is fluid in the droplet.
Okay.
Because that's bacteria, the active agents have to live in a fluidic environment.
so it's a fluid inside the bubble.
Because the droplet. Droplet means something contains water. So there is water in there. Yeah.
Two different colors for really two different density.
Sorry?
Two different colors.
Yeah, I think the bacteria is concentrated on the boundary, so it's like.
Bacteria is concentrated from the boundary. Start itching.
So is that it? Like,
I don't-
imaging I'm saying because.
I don't know what this is, to be honest, but yeah, I don't know.
But in terms of like, we already know that if there is a boundary, the bacteria will accumulate in it, okay?
Correct?
This case, and in the
Correct.
center it will be less.
Correct?
So we not think that at the center there is no density bacteria.
Because I don't know what's this imaging, so I can't say for sure. But physics, of course, we understand already, right? You have to look into the paper to see what kind of imaging I'm doing here. But yeah,
That's it.
good. Other comments? Okay, so we've seen a lot of systems, and in the last segment, we're just going to talk about a few examples and then discuss what we can do in the future and what are some of the challenges that we're facing. I'm sure you've seen that already in this lecture, in the previous lecture. A lot of things we don't understand yet. And I think that is the point of the workshop, not in early, first couple of days where we talk about dilute systems, where I can tell you almost everything about them. Of course, that's satisfying to listen to, but then the point is, what can you do with it? Not too much. Well, with these systems, there are a lot of things we don't know, and that is where you guys can make a contribution, okay? So one of the themes that we've seen through our lectures and discussions is these guys are active, so they're going somewhere, right? And their transport processes, meaning where they're going, their diffusive motion or their directed motion is important, both in biology, in nature, and in applications. For example, we talked about infection, motile bacterial infection. They have to go somewhere that we don't want them to go to cause infections. And we can also think about carriers. If we have an active carrier, that would be much nicer than a passive carrier, which is just diffusing around and gets wherever he wants to go, right? In the context of targeted therapy, we don't want that. We just want them to go where to the site that we want them. But of course, when we have activity with these carriers, the question is, how do we control them? We now have activity, but we don't know how to direct them to the places we want. And just in terms of general transport processes, there's a lot of interesting things that we still don't understand yet. So here's a very recent paper, for example, looking at a polymer made up of beads. Now, each of these beads are active. So this is a rigid connection. So they're not allowed to deform, as the simplest model that one can make. So this is a simulation work. So you have essentially just an active rod because they're not deforming, okay? But this rod has a interesting way of swimming. So it swims for a bit, and then it's just gonna swim in the opposite direction. That's a rule, not what happens in real life. So if you put that into a porous medium, and we want to understand where does it go, or what's the diffusivity, right? At long times, it's gonna move somewhere, spreading. And if you measure the effective diffusivity as a function of these parameters, this parameter compares so porous medium is made up of these pillars or obstacles that are fixed in space, and anything that moves through the medium can only move through the empty spaces. So you can define a typical size of these empty pores, right? And if you compare that to the size of the swimmer, notice there's an interesting non-monotonic behavior. So there's some kind of optimum geometry where these guys can spread the furthest away, so largest diffusivity.
Is being plot here?
Diffusivity as a function of the length scale comparison to the length scale. So length, there are two length scales. One is the size of these swimmers. Other is the size of the empty space in the porous medium. So there's an optimum diffusivity that you have depending on the geometry. Okay. This is simulations. Yes. Okay. So essentially, yeah.
How is that?
So you have here. You have a few particles. Each of them are active. So there's an active force pointing to the right on each of these beads. So this whole thing has an active force on it. So it moves around, just like the active Brownian particle, except that it's not one, it's a few that's connected together.
So, for example, let's say we had the Lagrange equation where there was one term,
Yeah.
it had US,
Yeah.
and then there was a WC potential.
Yeah.
So, WC potential can model your coordination.
Yeah.
Plus you have harmonic
Yeah.
for the backward.
Yeah.
And in that US, if you want to do that, you can do this F.
Exactly. Exact same way of doing that, except that you have to add these forces, additional forces, they're connected. That's right. Yeah. Other questions, comments? Okay, so if you think about these transport behaviors, they're dictated by their surrounding environments. And the question is, if you think about these things, either infection or carriers, they're often in a complex environment in the sense that there's flow, the geometry is non-trivial, and there are potentially other things in there, other particles that are going to crowd the environment, okay?
Sorry, what is happening in reverse?
Oh, that's a rule of the swimmer. So he just swims in this direction for a while, and then he's going to swim in the opposite direction. That's a computational model. You can do that in simulations. It's not a real experiment. Yeah. Ask these guys. I didn't do this work, okay? Other comments? So, okay, so there are a few things I've listed here that is for you to think about. There are many other factors we want to think about. One is complex geometries, right, as I've talked about. The other is fluctuating environments. So if you think about these biological channels and things, they're fluctuating. They're not a static geometry over time. So that's something we haven't talked about entirely here. So how do we think about that? When you have active particles, say, in a channel, but the wall is deforming over time. So that's very interesting. And that's something one can think about in the future. The third is crowding. And this we've seen before. Say in a cell, you have active particles in there, but there are many other things around there. How do we understand the transport dynamics within a cell? Okay, we talked about how motor proteins transport vesicles in the cell, and that's important for cell function. And the question is, can we understand to what extent does these active forces in there that promotes transport compared to just passive diffusion? We don't know. Okay, that's something one can think about. Any other comments or things to add to the list here that you guys think are important?
Crowding is look at the different types of active particles.
You can have, yes, you can have other active ingredients there as well, in addition to passive. We can also have
Tracers with a active particle.
tracers you've added, or
It's a crowding a lot of active particles that we.
a lot of, yeah.
It's like it's here against active system.
Yeah,
You've added the like a passive particle,
okay.
like different particles.
Yeah,
Or anyway.
you could be, and we're thinking about sort of applications, and what kind of applications are you thinking about?
Just thinking that the tracer can show around the text.
It does, yeah. Even in the dilute case we looked at, right? A tracer in a dilute active system, it picks up the activity because it gets kicked around. That's correct. Yep.
Okay. And so, right.
Other comments. So the other thing that
So that was what. So in the cloud, we have various systems dedicated, like let's say sustainable. So if the passive particles, like if you consider normal passive particles around the should have like this, is that problem is solved? What is that solution is adequate? Like
so you're thinking about purely passive system, or there's a lot of
say there is no active particle,
no active particles.
just normal drought of a passive particle.
Yeah.
So is that problem is solved, or do we have some inference from that problem?
People have done those in terms of simulations.
Can you think of a lot of S particles radiance? That's just the glass system.
People have done those things. Yeah.
Yeah, because as much as I can understand.
But that's the interesting part, right? So if you add activity, so we talked about one example before where we have a passive gel, and you embed a few active guys, you get new configurations that are not accessible, just for passive where it's thermally driven. So you will entirely create either new space in your face space, new behaviors, or some other interesting structures that emerge. Yeah, so those are something we want to think about as well. Yeah.
Yeah, I'd say polymorphs in manufacturing found more on.
Yeah.
With simulation that they, especially for DNA, that they try to see the solid specific by putting down.
This also with Excel.
Yeah.
Yeah.
They don't see. But that's not transcript dominant.
Oh, this is stupid.
Good. Other comments?
No, it's just
Okay.
yeah.
So active particles, they can transport payloads, but they're also pushing around other passive objects, right? They also generate flows. So, which means that, again, coming to your point about these passive systems, so here's a passive but sticky particles that's on the left. So there's a sticky interaction, short ranged. And if you put those in a bath of swimming E. coli, you're going to get thermally inaccessible configurations for these passive guys. And you can generate these new structures that's not possible without this activity. So that's something we want to think about is making use of these active guys. Can we design new materials and structures? This is experiments, and that are not possible before. So looked at the other case, again, the gel case where you can induce a brittle to ductile transition, things like that. So these work are only just getting started. This is 2023, okay? So there are a lot of things we don't know yet. For example, how do we predict the behavior? Okay, I put some bacteria in here, but what do I get? I don't know. And we need to develop models to predict that, whether it's simulation or some other principles that we can think about. So those are wildly new areas that we haven't done much yet in this field, which are interesting. Okay. Other comments about this? Okay, so we're talking about passive objects. In general, we can, even moving out of material science, in general, we're just trying to manipulate passive objects, with active particles. We want to be able to predict these structures, but also can we control their motion so that we can precisely guide them somewhere or these structures. So the point is then, can we design methods for automated control of particle motion, whether based on some sort of feedback control or some other things, external fields, forces, flows. We talked about chemical interactions before. Geometry, that's also something we've shown slightly. Okay, again, in this part, we need a few things that that can help us. One is really high fidelity modeling that incorporate these first principles of physics into the systems so that we can get quantitative predictions. That's really what we need in terms of designing these things. The other is we need to develop models in the spirit that we've done, cross-grading and things like that, at least give us a intuitive and physical understanding of the systems. And lastly, because there's so many degrees of freedom in the system, can we make use of some data-driven approaches to help us? If we just want to design something without want to go through the entire process of high fidelity modeling, understand all of the physics, can we still do that with a data-driven model? So those are a few things that people are thinking about. Here's a sort of a simple example of using machine learning, in this case, a reinforcement learning, to guide swimmers. So this is a simple, again, simulation model of a swimmer that's made up of 3 beads connected by 2 arms. So now this is put in water, and when we talk about swimming or locomotion, we have a gate, right? You take some sort of actuation, and you get a response from the environment, and then that interaction gives you self-propulsion. So in this case, the gate is simply, I'm going to allow the arms to extend or contract. And I'm also going to allow the angles to change, so I can change the relative orientation between these 2 arms. So if I put that kind of object that has the self-actuation in water, it's going to self-propel. And here the point is, can we guide them somewhere by tuning this gate over time through a machine learning model? And the way we do that is, here there's a video showing this kind of simulation. So let me stop this. And so you see this red dot here. I don't know if you guys can see that. So that's the location we want this swimmer to go to. Okay, we have that objective, and we feed that into this neural network by tuning the gate over time. Okay, so you can do that, and then you just update their target over time, because you're designing a path essentially. So you update this red location over time, and then one can see that the swimmer can go to these locations as you have designed. Okay, so you can write out swim over time. So here's a plot of the trajectory. So essentially you have a target direction somewhere in this configuration, but you started somewhere here at a different configuration. So what happens, the swimmer is going to take some time to transition from this configuration to a configuration that is aligned with the target direction. So there's a time scale associated with that, but eventually you can do that. So of course, this is the simplest case that one can do. It's just a single swimmer in a viscous fluid. Questions?
I didn't understand this, Jerry. So after learning, what is it doing?
It changes its gait, which is the length of the arms and the orientation, try to align with the target orientation.
Exactly. Yes.
Yeah.
So it's at the observation see it's learning about the system at a particular
So it takes a step, and then there's a feedback. It measures the step you've taken, is it good or not? Okay, and then it tunes it with this neural network, and then you will update it. And over time, you will eventually try to align with the target direction that you've specified. Yeah.
So it is like if it's favored to the direction of a target, then it
Yeah, you get a reward if you're going in the right direction, right? And then you try to go to that direction more. I think this is it's not a very complex model that they've done here, but essentially it's to demonstrate that this is possible for these kind of swimmers. Yep.
I think the difference with wanting other is that you are not just providing condition whether it is accepted or rejected. See, here I think the ocean is learning whether it's
There's no Monte Carlo that's happening here. We're actually taking a step, actually figuring out where we have come to.
That you doubt. So once the network is in the mechanics, once the network is built, then you can follow the code.
That's right. Yeah, exactly. So you can train them, and then
I think what she's trying to say is for training the loss function, we are using multiple optimization.
I don't think so. I cannot remember the second one, but I know that you raised Nigeria. No, yeah, something else. Yeah. Okay, so other comments? So that's what we talked about before already. You know, we can use these active ingredients to generate flow, especially at the microscopic level. So this is something I guess we haven't talked about. The microfluidic devices regenerate. We almost never use pressure to drive flow. Anyone know why?
What like?
So in microfluidic channels, which means channels that are very small, we know that we can, in regular channels, we can apply a pressure gradient. It drives the fluid flow, right? Yes? And then the question is, we normally don't do that in microfluidic devices. We use other approaches to drive flow, not pressure gradient. The reason is this is a sorry.
Like your security stuff on you is because it's sensing order of some microliter.
Yeah. Yeah.
So it's one is there like this destructive way as well.
Yeah, exactly. So the pressure very needed is tremendous, tremendous compared to large scale. So it's
not really
it's not useful. It's not helpful. So we can't do that. So we use other ways to generate flow. Yeah. That's the point where the potential, and this is interesting. We can use it to generate flow at the micro scale, but then, of course we can't do that now because we generate this flow is already useless. So how do we control so-called active turbulence so that we can get clean, steady, unidirectional flow, or maybe we want time-dependent flow, but we should be able to control that using some mechanism. So that's something that one can think about in the systems. Okay.
Normally, it's in our studies where that is flowing the capillaries.
Yeah.
There, so some of the macrophages track this way.
Yeah.
So, but we still don't.
There's two things. One is our blood is not Newtonian.
Yeah, that is not the American, but the channel where they are moving that capillaries that they are moving. And that is in the order of 8 by 2. That is considered domain of aqueous fluidics. So
Yeah.
So in that first level, there's some modeling regarding this how they are moving.
You could, yeah, yes. That's also very relevant. Yeah. Good. Other comments, suggestions on what we can do?
This is so interesting. Yes.
Okay, so this we've talked about already. Crowded environments such as in a cell, right? We don't really understand how these active force fluctuations benefits the cell, in what way and to what extent. And the cell has lots of other things in there, so those will modify the behavior. Okay? And the other thing that's kind of tricky is, we want to, if we do an experiment in these complex environments, we want to be able to look at these bacteria, right? We want to take an image, and we should be able to identify where they are. But if you think about porous medium, normally these optics are opaque. So to do any kind of optical imaging, you can't see inside. So then we can't directly track these bacteria. So that's not very nice. So here's a recent work where people have made these are hydrogel beads, which are optically transparent. So you can actually do nice imaging and actually can keep track of individual E. coli in this 3D porous medium. So that's very nice. And then you can look at where it goes over time. So that's interesting. So we need some sort of ingenuity in designing our experimental systems to be able to have more precise tracking, especially in 3 dimensions, okay, in this kind of more real environments that we want to understand. So here's sort of a classical setup of experiments, okay, just through optical imaging, and you have your experimental system in this microfluidic chamber, and you take images, and we heavily rely on image processing to extract useful physical data out of these systems. And speaking of image processing, because machine learning is everywhere, so people also have started doing that. So these are sort of raw experimental images, and then you transform them. Here you transform them again into a bright field image, which are much easier for later data processing in terms of extracting the shape and the geometry and the particle locations over time. So that's something that's also interesting in terms of future applications. Comments? Okay. So we also talk about these chiral systems. It turns out, in general, you can imagine chiral systems are more common, right? So non-chiral swimmers, which means everything is perfectly aligned, so they don't spin. But in general, of course, that's not the case. You know, in nature, in biology, things are not perfectly aligned, so they go around in circles. And here, this is a sort of emergent chiral behavior due to hydrodynamic interactions among different colonies. So you see that quite common. So when you add chirality, actually, there's a lot of things we don't understand. For example, in this case, we have chiral active particles. This is a simulation of chiral active particles, finite density, okay? And you can form these droplets. You have these traveling.
Again, this is iodide, not very dense. Like, isn't highly dense like
No, not highly dense, but yeah.
glassy?
No, not yet, no. So you have these new states that we don't get in just active Brownian particle systems. For example, you have these bands are actually traveling, and you have these droplets. Actually, the droplet in this case is rotating, but in the video it's showing just a droplet formation. This one is not rotating, but sometimes the droplet will rotate. I'm going to show a video in the next slide where it rotates, and you form these micro flocks. So these little guys are moving somewhere. So very interesting phenomenon, and that happens. Here's another simulation where once you form a cluster, so each individual particle is a chiral particle, active. Once you form a cluster, each of these guys are active, but they're also spinning, right? You can imagine this spinning motion sometimes reflect at a larger scale. So what you see is you get different kinds of states. One is this cluster is in a spinning state as a whole, spinning in a particular direction. And in another case, this spinning cluster reverses their spinning direction. So here's an example of that. This is again a simulation. So initially it spins in one direction, but then changes over time. And this is more or less regular, periodic over time. So that's quite interesting. So of course, this is not just a theoretical construct. If you look into biology, you have, so this is a starfish embryo that's allowed to develop in a petri dish. What happens in this case is these spontaneously self-assemble into a chiral crystal, and it spins, and it's quite persistent over, I think, long over more than 20 hours or something like that. Oops. Yeah. What's happened here? Good. Okay. So there's a clock here. This is a experiment. So this is already over 20 hours.
Just 20 hours in what simulation with that?
But in simulation, it's not real time, right? You scale things out. Yeah. So okay, so that's interesting. And these chiral systems really don't understand the behavior that much. Okay, so there are other things you can make active solids. So here is so you have these arms, but they're connected with a motor that can actually actuate these arms. So you can form these sort of meta materials structures, and they have locomotion. They can move around and they can climb these complex landscapes. So here's an example of what happens with these systems. Stereo compression experiment. And in this case, it's just moving in this direction, tumbling around. Okay. So there's a lot of things out there that we don't understand. And here's another thing that's interesting is in the context of using active particles for biomedical applications. So often in this case, we often want some sort of response to external stimuli. Often we want to use magnetic materials. That's the easiest to do. The other is we need, the other things for, say, delivery, we need cargo materials. And we also need sometimes X-ray contrast agents so we can track them. But a composite carrier or active deliverer carrier has only limited space. So you want to improve one aspect of this, necessarily you have to sort of reduce other part of it. So there's a sort of impossible triangle of designing these systems. So the question is, can we make use of these active things that maybe an active material can combine, say, 2 or 3 properties, and it allows us to resolve this tricky design triangle, right? For example, we have magnetic bacteria, which is already magnetic. That's great. You also can transport things. So that's something people are actively thinking about. And in the first lecture, we talked about how we can use magnetotactic bacteria for targeted cancer therapy in an experimental setting. So those are things that are people are thinking about at this point. There are other things. Again, this is related to the V-check model of intelligent active particles. So in the V-check model, you have a particle here, right? Say you draw a circle around this particle. I'm just going to align with everybody in the entire circle. But if you think about birds and things like that, they have a cone of vision. They're swimming in this direction. They can only see maybe swimmers in this cone of vision. So maybe let's just only align them with the particles in the cone of vision, okay? And that's all we're doing here. And if you do that, you get these strange structures, okay? And you see you form these rings and these bands here in the system. So again, that's something that we can think about. Can we? The other point is if you think about a swarm of drones, so that's something people use. For example, in some applications, can we make use of these rules? Can we build this physics into the drones and so that they can avoid colliding with their neighbors, and then they can have some coherent motion to do some task that one wants? Okay.
So that pattern is called whichever one of the different envelopes, like
which pattern here? Yeah, exactly. So the theta is half angle of the vision. 2 theta is the vision angle, right? So if you change that and change the speed of these guys, you get a lot of strange phenomenon that happens.
in this one, always wins. The extension of that.
You can view this as an extension or a generalization, where you take a form of vision in the V-check model and that would be high, that'd be the top, right? Something like that. Comments. You guys are very quiet now. I only have one more slide, so that's it. So last thing I want you guys to think about is cooperation. So in real systems, whether humans or ants, we cooperate, okay? And that's very important because sometimes the task we want to perform cannot be done by a single agent. So here's the well-known phenomenon for ants. They form these bridges in order to move from one place to another, okay? And the point is, this allows them to do collective problem solving, where a single agent cannot do that. Can we use that for our active particle systems and have them cooperate in a way, either for transporting some passive object or to perform some tasks that we need them to do? Okay, so that's another thing that we want to think about. And this is completely, we haven't thought about this at all at this stage, but that's something, you know, we see in nature. Question is, can we borrow that for these applications? Okay, so with that, we have gone through our entire program, and I'm done with my lectures. If you have any questions, feel free to ask. And then in the afternoon, we also have a few tutorials where we're going to continue with the simulation that we're doing yesterday. And tomorrow, we're going to solve some analytical problems. And that is the end of the lectures. Okay, thanks everyone. Any questions? Okay, there's still a few minutes, so we can, if you have questions, you can ask. If not, you are free to go.
Good. Is that stand limit in this is
The beehive probably those who are similar collective probably.
Yeah. Questions and there is something on them. We're straight up and down. I guess you could. You don't have to down the atoms. You just go out there and watch them. Each of them to see one of their. Yeah.
yeah, the sunshine. This was the
It's the afternoon, pretty spring your laptops, you know what I mean? So I can do installations.
attitude.
See, on the channel.
It's a sound. What
Feels like you go on when I'm like it's a me.
Yeah,
What he did with you.
was
I should.
Yeah.
What's the ocean?
Definitely.
Getting safe and low to that one things.
That's it.
All you Get me storing this. Yeah. You think of that.
You could. Yeah. But I don't dance.
I was scared.
Sitter.